Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. And hopefully everyone is doing fine. So as I promised last week in our class, so this one is the continuation of the microscopy system known as AFM. Abbreviation is AFM, atomic force microscopy. So why is the name is atomic force microscopy? Let's look at this diagram. Okay, so this one is the sample surface, and this one, the blue color one with the sharp tip, is called cantilever, and the tip, that's the cantilever and the tip. So this tip will be scanned across the samples, right? So how do they acquire the images? So there will be force applied on top of this cantilever. That means this cantilever tip will have a force amount at atomic level, and then the positioning of this laser beam on top of the tip, cantilever tip, will be used as a reference, and also intensity and also topographical what we call information data. So the laser beam is positioned on the tip across when the sample is moved across the sample. So with the atomic force, it will be able to tell the distance between the samples and the tip. That is being measured, and the deflection of this cantilever tip is measured when there is a difference in the positioning of the lasers that's been reflected from the back of this cantilever tip, and it is measured through the photodiodes, or what they call, they use this as ray tubes, which is will keep on getting the signals and the changes that happens, and this will be used as the recording for the data. And this is called the detectors and feedback in electronics. That means sometimes the AFM can be an advanced system, where the one that I'm explaining now is the basic of AFM, but later on we will look at a few example of imaging mode that can be used in the AFM imaging. So whenever there is some sort of close contact between the sample and the cantilever tip, this will be sent as a feedback. So will be sent as a feedback to the system, so that the cantilever will be adjusted automatically, so that it will not ruin the samples. That means the samples will be safe. But that is in the advanced mode of AFM. So the one that we are looking at now is the basic AFM system. So by scanning a small, what we call area in the sample, let's say for example 20 micrometer times 20 micrometer, you will be able to tell the topographical information between the square that's been scanned over the surface sample. So this is happening by the tip is bending over the on top of the samples. So as I mentioned, in the system, AFM system, there are three modes that can be found, which is called the tapping mode, contact mode, and also non-contact mode. So what are the difference between this mode? I'll explain to you that. So if you can look at this diagram, so it shows you that, let's say this is the sample, then you have this cantilever. So the cantilever will be moving across the samples in the tapping conditions, so that when it reaches to the sample, it will also adjust the cantilever height so that it will not damage the sample. So it will not damage the sample. That means it will move in the raster scanning as well, but when it comes to an object, it will start to adjust the cantilever height so that it will not scratch the sample. So that's the tapping mode. Another one is contact mode. So you can get an accurate or high resolution images, but the contrast of this the disadvantage of this method is that you will potentially damage the samples, because the tip will be run across the samples. So it is not like the tapping one previously explained. So it will go across the samples. So that's called as contact mode. Another one is non-contact mode. Non-contact mode is what we call permanently positioned, that it will not be in contact with the samples, and it is not also the tapping mode. So it is scanned quite far away from the samples. The cantilever is positioned quite far away from the sample. So by comparing these three methods, you will be able to tell that which one is the best method. So in non-contact mode, since the cantilever is far away from the samples, so you are most unlikely to damage the samples. But in contact mode, the sample will be damaged, but in tapping mode, your sample is also still safe. So that's the three modes that can be explained in the surface plasmon system. So to summarize the AFM mode operation, so this is the table of modes of operation that you can digest for the system. Is start with the modes of operation, working principle, advantage and disadvantage. So each of the system that we the methods that we mentioned has its own advantages and disadvantages. So for example, contact mode, so the working style is principle is a physical contact between the tip and the surface. That means is obviously the name is in contact mode, right? So you will have contact with the samples. Advantage, high speed scan, high resolution, because the tip is quite close to the samples, or ultimately touching the samples. Disadvantages, damage to the soft sample. Later forces may produce image artifacts. I mean the scanning after that you will do may produce the image artifacts. Followed by non-contact mode. So non-contact mode, you will have the no contact between the tip and the samples. So it is positioned quite far away from the samples. Low resolution, because the tip is positioned far away, so low resolution. No damage to the sample. No damage to the sample. Disadvantage, slower scan speed if compared with both contact and tapping mode. So that's the disadvantages. And then if you look at the tapping mode, it's intermittent and short contact between the sample and the tip. That means short and intermittent, the contacts. Advantage, you have high resolution, minimal damage to the sample, slower scan speed if compared with contact mode. The speed is slower. So each of the system has its own advantages and disadvantages, but between these three, you have to identify which one is most suitable for your samples. In, for example, if you are scanning tissue samples, or the fabrication. For example, from the last lecture, I've mentioned to you about the patternings of protein micro contact printing system. So if you have extracellular matrix protein, so in that study also I have used the AFM system, and you have looked at the images. So based on these three, which one? I'm sure you can tell your answer in the comment section. So type your answers, which one will be the most preferable one, and why do you think that is useful to be used in the surface patterning technique for this AFM system. Okay, let's look at this figure. AFM morphological images. That means you can understand from the word morphological means that we're trying to look at the morphology of the samples. So sample A, B, C. Sample E is the solvent casting, poly, PCL films. Scan area is 50 times 50 micrometer. B is PCL gel, electrospun membrane, scan area 20 times 20. And then C is PLGA, electrospread microparticle, scan area is 20 times 20. So can you tell the difference? So the 20 times 20 is much better in the resolution compared to the 50 times 50, and also this has to do with the types of material deposition. So this one is the types of studies that they do for structural properties, whether to study the material properties. For example, in building a scaffold, if you're building a scaffold for human tissue replacement, tissue engineering, so you can use this system to identify the scaffold orientation, scaffold what we call porosity, scaffolds surface properties, whether it is suitable for cell seeding, so that the cell will grow into the scaffold. So this one is the types of AFM images that you can produce when you use a different size of scan area. So A is 50 times 50, B is 20 times 20, and C is again 20 times 20. So before we have look at one of the examples on building the scaffold, so this one is another example of AFM based nanoscopy of living cells. So AFMs, since they works by sensing the top surface area, so you can tell that there is a cells, and then you have the strands, the membranes, and then the internal features of the cells. So how the cells, the cantilever is positioned on top of the cell, so that you can get close to the cells to identify the cell attachment properties. That mean how the cells actually position on the surface, and by doing the complete scan, so you can look at the surface topography of the cells, whether the cells in terms of cell position, cell attachment, and also cell intensity, so how they interact on the surface with the different molecules, strands, proteins, and DNA. So this is another example. So I think that's two examples given for AFM system, which is you can explore further. So I think that's the two examples given for AFM system. One is on the scaffold, another one is on the cell engineering. So there are many other applications on biomedical engineering, such as for tissues and cell engineering, and also in food science, food technology. So AFM system have been used widely, and also you can go back to the same papers that I have explained previously in the wide field surface plasma system. If you can compare the advantages and disadvantages of both the system, that means what are the speciality of each of the system. we cannot say that AFM is better than WSPR and WSPR is better than AFM, because each of them, they have their own advantages and capabilities. So, and they can complement each other in the research wise. So what we like the student to do is answer the question that I've asked earlier, and also please find out other studies and application and the advantages of using AFM system in imaging biological samples, biomedical samples, so that you gain an understanding on how the system functions, system works, and why they use the system. So hopefully this will be a good task to explore. So hopefully you will take it seriously and complete the task, and see you inshallah in the next lecture. Wassalamualaikum warahmatullahi wabarakatuh.